Hello, good evening. It's wonderful to see all of you here tonight. I'm Gina Vild. I'm the Associate Dean for Communications and External Relations at Harvard Medical School. It's my pleasure to welcome you to this third Longwood Seminar of 2017, From the Wings to Center Stage, How Inflammation Triggers a Multitude of Diseases. When we asked our Longwood Seminar audience to vote for this year's topics, this discussion was at the forefront. Inflammation is one of the body's first lines of defense against disease. But as you likely know, there's a growing evidence that chronic or acute inflammation can be a catalyst for a host of health concerns. Autoimmune diseases, neurodegenerative conditions, heart disease, to name only a few. Tonight, HMS researchers and clinicians will explore the connection between inflammation and chronic disease. And our goal is to ensure that you leave here with a good understanding of the subject, and we all leave here with an understanding of how we can reduce our risk. So before we begin our program, I want to formally welcome all of you here in the auditorium, but also those watching from live streaming. I thought you'd be interested to know that 2 weeks ago at the last Longwood Seminar, we had viewers from 58 countries. From Vietnam, Kuwait, Serbia, Nepal, Somalia, Algeria, Poland, South Africa, Lithuania, Hong Kong, to name just a few. That represents viewers from 30% of all the countries in the world. So that's quite remarkable. In addition, the last seminar reached 288,000 people through Facebook alone. So to those of you here in the audience, I'm delighted you could be here, and I equally want to welcome those of you watching from live streaming. So just a few brief announcements. If you want to view past seminars and have access to the supplemental reading material, you'll find it on the website. There's a slide that should be coming up that will give you some additional information. You're probably aware you can receive a certificate of completion if you attend all 3 of the 4 seminars, and teachers can receive professional development points if they attend all 4. And there's information on how you can do that. And last, please do not turn off your phones, contrary to what we generally say, but leave your phones on and just silence them, because we're really hoping that you will join the conversation through Twitter using hashtag HMSMiniMed. And we invite our viewers through live streaming to also send us your questions for the Q&A at the end of the session through Twitter. So now our panelist. Our moderator tonight is Dr. Diane Mathis. She's a professor in the Department of Microbiology, Immunobiology at Harvard Medical School, and holder of the Morton Grove Rasmussen Chair in Immunohematology. Dr. Mathis is an associate faculty member of the Broad Institute and serves on advisory boards worldwide. Her lab focuses on T-cell differentiation, autoimmunity, and inflammation. She has trained well over 150 students and postdoctoral fellows. We're delighted to have her moderate. Dr. Filip Swirski is a Scott and Patricia Eston MGH Research Scholar, Principal Investigator at the Massachusetts General Hospital Center for Systems Biology, a faculty member of the Harvard Immunology PhD program, and an associate professor at HMS. His lab utilizes a range of cell, molecular, and imaging techniques to elucidate how innate immune cells contribute to cardiovascular disease. He focuses on hematopoiesis and monocyte differentiation. Dr. Beth Stevens is an associate professor at Harvard Medical School in the F.M. Kirby Neurobiology Research Center at Boston Children's Hospital. She's a member of the Broad Institute. Her research seeks to understand the mechanisms that regulate the development and elimination of synapses by focusing on how microglia and immune-related molecules mediate this process. She's investigating the mechanisms that drive synapse loss and dysfunction in AD, Huntington's disease, as well as neurodevelopment disorders such as autism and schizophrenia. And finally, Dr. Stephen Shoelson is an Associate Director of Research and Section Head of Cellular and Molecular Physiology at the Joslin Center for Diabetes. He is a professor of medicine at Harvard Medical School. Dr. Shoelson co-founded Catabasis Pharmaceuticals in 2008, where he also serves as the chairman of the Scientific Advisory Board. He's also a member of the Scientific Advisory Board of Metabolics and Genmedica Therapeutics. He has published over 130 papers and is considered a leader in the field of diabetes signal transduction and inflammation and insulin resistance. Please join me in welcoming our esteemed panel. Thank you, and please do stay for the Q&A. Dr. Mathis?
So good evening, and thank you for coming. I think I speak for the organizers and all of the panelists in saying that it's really amazing to us and inspiring to see that there were over 400 people that expressed an interest in learning about inflammation. So I'd like to start things off by giving a short introduction to provide some context for the three research presentations. So what is inflammation? Inflammation is the body's response to tissue injury, whether it be by microbial infection or chemical irritants or mechanical damage. When it begins, it's primarily a response by the vascular and immune system, but eventually many other types of cells are called in to the site of action. And throughout this response, the various cells which are engaged produce a series of molecular mediators that have very precise roles, and they come into play over time. The function of inflammation is basically threefold. Number one, it gets rid of the irritating stimulus. Secondly, it manages to clear dead cells and other tissue substances that are at the site. And third, it starts the repair process in action. So I think every single one of you here has experienced the cardinal symptoms of inflammation. Oops. Where's the pointer? Bottom. Okay. I think all of you have experienced these symptoms. First of all, what happens is that there's heat and redness at the site of injury, then eventually swelling, and then pain, and often, although not always, loss of function, which is quite often due to scarring and scabbing. So these features of inflammation were recognized many, many, many years ago, even by the Romans. And so medical students still today learn the Latin terms for these different stages, which are calor, rubor, tumor, and dolor. And I think it's interesting to keep these in mind because some words that we have today still use these Latin terms and reflect the same processes. So, for example, calorie is to do with how much heat is generated. Tumor is a swelling that shouldn't be there. and dolorous refers to mental pain. So underlying these external symptoms is a whole highly orchestrated set of cellular processes that take place. So when the injury occurs, cells at the site, in particular cells called macrophages or mast cells, are able to sense the injury. Either they can sense molecules at the surface of or secreted by microbes, or they can sense molecules at the surface of or secreted by damaged cells. So when they sense this damage, they become excited and produce a number of different molecular mediators. So some of the mediators, one set of mediators, is very important because of its effects on the vascular system. So these are things which you might have heard of, such as histamine, which is made by mast cells, or serotonin, or bradykinin. And what these do is to dilate the blood vessels, causing an increase in the blood flow. And this is what causes the redness and heat, which I described. And another thing that happens is that the vasculature becomes leaky, leaky to both liquids in the blood as well as cells coming through. And this leakiness leads to edema, and the edema eventually leads to swelling and pain. So that's one side of the story. The other side of the story is that these cells produce another set of mediators which are important for calling in a whole bunch of other helping cells. So the number one cell that comes in is a neutrophil, and it's very important for cleaning up the mess that's there. Others, and also producing additional mediators called cytokines. So other cells come in, such as precursors of macrophages, eosinophils, and eventually their combined activities lead to tissue repair. Now, if the irritant can be removed, and if repair takes place relatively rapidly, this is called acute inflammation, and involves this set of cells, these phagocytes that I mentioned. If the repair doesn't take place, or it can't get rid of the irritant, chronic inflammation can set in. And what happens then is that a whole different sets of cells come in which fight an immune response here. So there's acute inflammation, and there's chronic inflammation. And inflammation can be a very positive thing, getting rid of an infection or repairing damage to the tissue. But it can also be a negative thing, especially when it's chronic, because that leads to constant activation and fibrosis and tissue damage. So I just wanted to emphasize a little bit the differences between acute and chronic inflammation. So I already mentioned that the stimulant for acute inflammation was microbial pathogens, or chemical irritants, or mechanical damage. Now, the stimulus for chronic inflammation is often acute inflammation that just doesn't resolve. Another stimulus is an autoimmune reaction, like in multiple sclerosis or type 1 diabetes. The cells that are involved are overlapping, but there are some that are distinct. So in acute inflammation, very important are these neutrophils, which clean up things that I mentioned, and especially macrophages. Macrophages are also important in chronic inflammation, but the adaptive immune system, or an immune system which is able to make response against particular antigens, also comes into play in chronic inflammation. So as the names indicate, acute inflammation is immediate and lasts over a scale of days, whereas chronic inflammation is delayed and can last months or even years. And then finally, the best outcome from acute inflammation is resolution. But there can also be, if things weren't regulated properly, or if the stimulus is difficult to get rid of, there can be abscess formation, or chronic inflammation will eventually set in. And the outcome of chronic inflammation is tissue destruction and fibrosis, which leads to scarring. Okay, so you've heard a lot, many of you perhaps, about inflammation over the last several years. We've known that inflammation was important in a lot of different diseases. For example, autoimmune diseases like multiple sclerosis and type 1 diabetes. It's been known for many years that inflammation was involved in these diseases. Also, arthritic diseases or bone diseases, such as osteoarthritis or osteoporosis or rheumatoid arthritis. It's known that inflammation was involved. And diseases like asthma and COPD, chronic obstructive pulmonary disease, were also known to entail inflammation. What's new over the past several years is the increasing recognition that chronic inflammation plays a role in initiating or exacerbating diseases of a number of different organ systems. For example, cardiovascular disease, so in particular atherosclerosis or stroke, metabolic diseases such as type 2 diabetes, fatty liver disease, and neurological diseases such as Parkinson's disease, Alzheimer's disease, even some indication for autism. And that's going to be what the focus of the rest of the speakers will be this evening. And so I'd just like to place them here. Phil will be the next speaker. Oops. Phil will be the next speaker, and he's going to talk about inflammation in cardiovascular diseases. Steve will talk about metabolic diseases, and then finally Beth will talk about the role of inflammation in various neurological diseases. So thank you, Phil.
Good evening, everyone. So for the next 15 minutes, I would like to discuss with you some of our evolving understanding of the role of inflammation in cardiovascular disease. So let me start with the good news. The good news is that the rate of death from cardiovascular disease has actually been declining over the last 50 or 60 years. It's not been a very sharp decline, but we've seen fewer people, proportionally speaking, dying from cardiovascular disease. And this has been correlated with some of the scientific and surgical discoveries that have happened. But of course, correlation is not causation, and you can overlay this decline with improvements in public health policy. For example, laws restricting smoking in public places. So that's the good news. But the bad news is that cardiovascular disease remains the #1 killer in the world. So this is not just in the Western world, where we have foods high in cholesterol and fat. Worldwide, cardiovascular disease is the #1 killer. So what is cardiovascular disease? When you think about cardiovascular disease, what does that mean? Well, there are many different types of cardiovascular diseases, from various cardiomyopathies, heart failure, traumatic heart disease. Many of you will recognize some of these as diseases that you might have suffered or your loved ones have suffered. There is a tremendous number of things that can go wrong with our heart. We don't know everything about these diseases, but we know a fair bit. So just to illustrate, this is a textbook called Heart Disease. It is a textbook that's essentially required reading for any student of cardiology. This is a heavy textbook. It's 1,000 pages of thin print, 50 chapters, and it covers many of the diseases, or an evolving understanding of many of the diseases that you see here on the screen. And it's updated every year. In fact, that's not the entire textbook. There are 2 volumes, 2,000 pages, over 100 chapters of the various types of things that comprise, broadly speaking, cardiovascular disease. But for this talk, I would like to focus on one particular disease that is on this list for a very important reason. It is a disease, a process, that in many ways is the cause of what causes most deaths. It is in many ways the cause of myocardial infarction, so a heart attack, or of a stroke. And this disease is called atherosclerosis, or hardening of the vessel. So what does that mean? What does atherosclerosis mean? So here we have a human and the vessel system in the human. There are arteries, those are the ones in red, that deliver oxygenated blood to all the tissues. This is the blood that goes from the heart and the lungs and feeds all your organs with oxygen and nutrients. And we have the veins, these are the ones in blue, which return the blood back into the heart and to the lungs. So that's the circulation of the blood. Now, if you look at an artery in cross-section, and you just sort of slice it open, you look at it in cross-section, a healthy artery looks something like this. What you have, it's a pipe, it's a tube. Inside here is where all the blood flows. And there are 3 layers that sheath this tube. There is an internal layer called the intima, which is really the contact between the blood and the tissue. There is a medium layer called the media, and this is comprised of cells that are very elastic. So this imparts on the vessel the property of being able to respond to blood pressure and the beating of the heart. It can squeeze and it can relax. And then there's the outer layer called the adventitia, which imparts structural integrity on the vessel, among other things. And this is what a pretty artery looks like. Unfortunately, for more than 95% of us, in some regions of this artery, in some cases as early as the 2nd year of life, what happens is fat accumulates in this layer, the intimal layer, cholesterol, LDL cholesterol. You might have heard of it as bad cholesterol. Different types of lipoproteins accumulate in that vessel wall. And it is a process that is chronic. It can last for years. Over time, these plaques, or lesions as they are called, grow. As they grow, they limit the amount of blood that can pass through there. The lumen, this area, shrinks. And they keep on growing and keep on growing. And what happens is that they can rupture. And when they rupture, all that compartment that's inside, in between those 2 layers, meets the blood. And when it meets the blood, what you have is coagulation. What you have is a body's defense system to stop bleeding. And when you have coagulation, you have blockage of the vessel. No more blood can pass through. And when that blockage occurs in the vessels that are feeding your heart with oxygen, this is when you have a heart attack. And if that blockage occurs in a vessel that is feeding oxygen to your brain, this is when you have a stroke. So this is a very dangerous, catastrophic complication of atherosclerosis. And for many years, since atherosclerosis was recognized as a phenomenon, it was just thought to be this: the slow accumulation of cholesterol in the vessel wall. But if you put a little microscope into this area and look closer, what is in there? You see that there is a bustling activity. Though every single of those blue dots that you see there, so this is the lesion in cross-section, every single dot there, that is not fat. That is a cell nucleus. These are cells that should not be there that are there. Every single green dot is the same cell that is very active. It's replicating. They're self-dividing. There is a bustling activity of cells, not just cholesterol. And we know a little bit about what kinds of cells live there. So every red that you see there denotes a cell that is called a macrophage that resides in the intima in the growing lesion. And there's plenty of macrophages that are there. And macrophages are certainly not the only cells that live or that can be found in lesions. This cartoon, just in a different perspective, demonstrates all the different types of immune cells that can be found in a growing lesion. From T cells to macrophages to dendritic cells, the list goes on. There's a whole community of cells. And so now you can ask yourself two questions. The first is, how do they get there? And the second is, what are they doing? So let's start with the first question. How do they get there? One thing we know is that nearly all immune cells, they're sometimes called leukocytes, white blood cells, they are born in the bone marrow. So the bone is not just there to sustain us anatomically, but inside the bone, there is a very complex architecture, locations called niches, that are home to hematopoietic stem cells that reside, that stay with us for our entire lives. These hematopoietic stem cells are coddled in these particular regions in the bone, and what they do is by proliferating, by self-replicating, they give rise to all our red blood cells and most of our white blood cells. And here is just a demonstration of these little hotspots. These hotspots are locations where there's a lot of activity and a lot of production of immune cells in those niches. These cells that are born in the bone marrow leave the bone marrow, and they do this all the time. And that process is a very active process where molecules that attract cells out of the bone marrow sequester them into the blood. When they enter the circulation, that's where they can start infiltrating lesions. But leukocytes are not the only ones that can enter the blood. Stem cells can do it too. And they do it on a daily basis. They do it over the course of a day. When you're asleep, they come out, then they return back. It's a circadian pattern of stem cell exit and return back between the bone marrow. But under some conditions, stem cells can settle in so-called extramedullary organs. Extramedullary because they are outside of the bone marrow. Spleen is one example of such a place. And when they do so, they can give rise to more of these immune cells by a process that is called extramedullary hematopoiesis. And these cells can then return back to the blood. And this is a movie of such a cell. Let me see if I can get it to work. I think so. This is a movie. Now, what you've got here is the blood is going this way. Green is an immune cell. And blue is just we're tracking its path over the course of 1 hour. And this cell is, I'll just play it again so you can have a look. This cell is residing outside, and it can slowly migrate. And when it finds the vessel, It can intravasate, and then whoosh, it goes out into the body. So now they're in the blood. What happens next? Well, what happens next is that they can accumulate in the vessel wall, in those lesions, because those lesions accumulate your cholesterol. What happens is with excess cholesterol in the blood, that cholesterol accumulates in the vessel wall. Sometimes it becomes crystallized, and these cells, these immune cells, follow those lipids. They follow those lipids by first migrating across that first layer, the endothelium, which is a layer that is a first barrier. And once they're inside, they can become big bloated macrophages, which continue to eat this cholesterol, which replicate. And I'll show you a demonstration of such a macrophage, what it can do when it sees, for example, cholesterol crystal. So here is a movie. These are macrophages residing here. And the black stuff is cholesterol crystals. And look at how they sop it up. As it comes here, they just eat that stuff. They just eat it all. Because these are cells called phagocytes that have been discovered over 100 years ago. Phagocytes means big eaters. They have evolved to eat. This is one of the things that they love to do the most. And they eat and eat and eat. And as you have more and more of that cholesterol, they keep on eating. They become foamy cells, and when this happens, they can't stop. What happens is that they may die. But before they die, they can cause a lot of damage. They can produce all sorts of enzymes and all sorts of molecules to attract more of these cells. And when they die, they cause an area of dead tissue that is in some ways inert, and it can be very dangerous in terms of, insofar as it can promote this plaque rupture. So I'll leave you with two more thoughts. One thought is that we should not think about atherosclerosis as a local problem. This is a problem that affects the entire body. There are events that happen in those lesions, but those events may lead to, in this case, the growth of lesions, the occlusion of a vessel after thrombus, myocardial infarction, which is heart attack, which then can act on the brain as it senses pain and reactivate the bone marrow to produce more of these cells. There is a vicious cycle here. That's the first thought. The second thought is, well, why are these cells doing this? Why are they coming in there, eating all this fat, and then just attracting more cells? Why not stop? Well, they're doing something that they have evolved over millions of years ago to do very well. Think of a blister, like a splinter, rather. My son had one of these yesterday and did not want to get it removed. And what happens, as you all know, over the course of several days, unless it's removed, what you have is you have swelling, as Diane mentioned, you have pain, you have festering. That is inflammation. Those are macrophages and neutrophils coming in. Once you remove that splinter, after a few days, there is no evidence of an injury. In atherosclerosis, there is continued accumulation of that cholesterol. And that is why a process that is actually beneficial can backfire in the event of non-resolving persistent inflammation. Thank you.
Good evening. So I'm going to take over from Filip, and I'm going to switch a little bit and talk now about type 2 diabetes instead of atherosclerosis, although some of the data I will present to you will show you how closely linked those two can be in terms of the underlying pathologies that promote them. They seem quite different on the surface, but deep down, they are very similar. So diabetes has gotten a lot of press. It is, there's an epidemic of it going on, and trying to understand what causes it has, next slide. Inflammation, also on the cover of one of these magazines, mentioned some of the diseases that are thought to have some involvement of inflammation, including heart attacks, cancer, and Alzheimer's disease on the cover, and type 2 diabetes was mentioned in the text here also. So it's coming to your attention. Now, before digging too much deeper into discussing diabetes, I thought I'd discriminate the two main types of diabetes for you, type 1 and type 2. One of them is an autoimmune destruction of cells that produce insulin. So that's the body's own immune cells attacking the insulin-producing cells. Whereas the other type 2 diabetes, does not have that at all. And it is much more associated with weight gain and a promotion of insulin resistance and the inability to produce enough insulin to overcome the degree of insulin resistance. Type 1 diabetes has historically been thought of as being more prominent in children, so it was called juvenile onset diabetes, whereas type 2 has been seen more frequently in adults and is called adult onset, although we increasingly see more cases of type 2 diabetes these days, and that's probably because we see more cases of childhood obesity these days, and obesity is a major promoter of type 2 diabetes and cardiovascular disease. So type 1 requires insulin, type 2 does not. About 5% only of diabetic patients have type 1. So although we care about them a lot because they're usually young people, it's a small number, whereas about 90 to 95% have type 2. And the prevalence of both is increasing, although type 2 diabetes is increasing most rapidly. And I will present you with some, this is getting a little bit old now, but it certainly tells the point, and this has continued, so that the prevalence of obesity seen in these maps of the United States has increased with time. You can see as the colors change. So much more obesity over the course of these slightly over 10 years, much more obesity. And similarly, there has been a very rapid increase in the prevalence of type 2 diabetes. In fact, they look like they're happening at very much the same rate, suggesting to us that there's a very strong connection between those, and we know that there is. And that is what inspired many of us to look for the pathogenic mechanisms, and that's where inflammation came into play. So we're not the first ones to think about this connection. Actually, a guy called Ben Franklin thought about it a while ago. To lengthen thy life, lessen thy meals. So don't eat as much, and you'll probably be healthier. And that remains just as true today as it did back then. And that it is the obesity, we think, that promotes a lot of the inflammation, and I'll try to explain a bit of how that occurs. But first, I just wanted to tell you, so insulin resistance is a concept. It's not very well understood by a lot of people, but what it means is that at the same amount of insulin, your cells, if you're insulin sensitive, they do respond, whereas if you're insulin resistant, your cells don't respond very well. So although you're making insulin, your cells aren't answering the call. They're not waking up and doing what they're supposed to, like take up glucose. So the pancreas produces more insulin, and that helps, and more insulin, and that helps. But at a point, the pancreas can't keep doing it, and it starts to waver. And the amount of insulin it produces is not going to be enough to overcome the degree of insulin resistance, and that's when diabetes occurs, in type 2 diabetes. So I've just put up here a few of the milestones that we think of when considering inflammation as a hypothesis of a mediator of diabetes. Epidemiologists were seeing back in the 90s and even before that, there were elevated levels of circulating inflammatory markers and mediators in the bloodstream of patients of subjects that developed diabetes or that were obese. Cell biologists showed that some of those markers in the blood could actually, in growing cells in a Petri plate, could cause those cells to respond less well to insulin, suggesting they had insulin resistance. So maybe these cytokines, these inflammatory mediators, could cause insulin resistance, was the hypothesis. So I'm giving you different styles of experiments because each one gives us different information. Epidemiology tells us a lot, but it never tells us causation, whereas a clinical trial might tell us when an intervention is going to help. So some clinical trials were initiated to try to see whether or not these would reduce the burden of type 2 diabetes or even prevent it from occurring in people that might be predisposed. So there's 2 clinical trials listed. One is a very old anti-inflammatory agent called salicylate that's been used for many years to treat joint pain. And the other is a newer type of strategy where we target IL-1 beta, it's called here, but we're targeting specific cytokines with neutralizing antibodies or other kinds of blocking molecules. And the other major finding was that just as Filip just described in the plaques of an atherosclerotic in the atherosclerotic plaques, that immune cells aggregate in sites of inflammation, but in this case, where you gain weight is where we see them, and that's in the adipose tissue itself. So but first, I'm going to take you back to the epidemiology, because this is a kind of a study where many people are involved in participants in the study. In this case, almost 28,000 women from the Women's Health Study that did not have diabetes or other diseases at baseline were followed. And over the course of 4 years, 188 of those, nearly 28,000, developed diabetes. And then these investigators asked the simple question, what was different about those that did get diabetes from those that didn't? So the ones that didn't are the case controls, and they took 2 times as many of those, so they had 363 that were matched. And what they found was that there was an elevation level of 2 different inflammatory markers or mediators in the blood of these women, something called CRP and another molecule called IL-6. So they suggested those predict the onset of type 2 diabetes. And there's what the data look like. So on the left are just the insulin levels, and they're twice as much in the people that got diabetes. Now that's a little bit of a paradox. You figure that diabetes is associated with too little insulin, but these are insulin-resistant type 2 subjects, and they have elevated insulin, and yet they're the ones that became diabetic. Not enough insulin. Also, elevated at about the same amount are CRP and IL-6. So these aren't great elevations. These are relatively small. In the kinds of inflammation where you've got an infected finger, you're going to have elevations of 100-fold or 500-fold in some of these kinds of inflammatory mediators. We don't see that in this chronic, indolent kind of subacute inflammation. But they're real, and they're very reproducible, so we're investigating them. Here's a picture on the top, across the entire top. Now, this is a picture up close of adipose tissue. And each of those big green blobs is a single fat cell. You can see on the left, and we're just staining the lipid in the fat cell. In the middle, on the top and bottom, are cells stained for their immune cells, in this case macrophages. And then on the right, we're looking at both types of cells. On the top, it's from, in this case, a lean animal, and on the bottom, it's an obese animal. And you can see the difference is that there's significantly more cells surrounding, in this case, is one adipocyte that we're focused in on, but we can see this across the entire expanse of the adipose tissue, that there's a lot more immune cells in the tissue and in specific areas of the tissue when obesity has occurred than when the animal remains lean. So that's a correlation, and we have studied many of the different mediators that have been produced, and this is kind of an idea of what might be happening. So the adipose tissue expands in obesity, and many of these cells accumulate there, and they produce substances, as both Diane and Filip suggested, and those substances go into the bloodstream. The same thing can happen in liver. So it also swells with fat, and there is an induction of some inflammation in the liver as well. So we think that those tissues themselves can become insulin resistant, but they can cause insulin resistance also in distant tissues like muscle, and shown here the kidney, and probably similar pathogenic pathways are activated in the vasculature and promote the atherosclerosis that Filip had mentioned. So the clinical trials. Here's a result of a clinical trial being done in patients with type 2 diabetes, and this is an IL-1 beta neutralization. So this cytokine is elevated in patients that have diabetes, and they treated them with the neutralizing agent, and you can see that the blood glucose came down. There's a fasting blood glucose level and a hemoglobin A1C level off to the left. And the plasma glucose level is shown on the right. So reduced in both cases, on the left and the right. And the same thing can be shown with salicylate, this old anti-inflammatory molecule that's been used for many years to treat joint pain. We can see fasting glucose on the left going down, and there's 3 different doses of drug shown here for these placebo-adjusted data. And then on the right, the hemoglobin A1C. And the difference between fasting glucose, that measures your glucose level at a moment in time, and hemoglobin A1C, which allows us to get an integrated value of blood glucose over, let's say, 6 to 8 weeks. That's a difference, but they both respond nicely. So we know that we're getting immediate effects and long-lasting effects by treating the patient with this drug. So there are other bigger trials underway now. The type of clinical trial matters and how much we put our faith in it. There are at least 2 very large clinical trials ongoing now with thousands of people enrolled. They'll be in the trial for years, and they'll be followed and treated with a drug against placebo for years. So these trials cost hundreds of millions of dollars to know whether a drug helps in the case of cardiovascular disease. But nestled in, because we think the pathogenic mechanisms are very similar, nestled into both these trials, the primary endpoint is cardiovascular disease. Nestled in a secondary primary endpoint is diabetes. So we're going to get 2 answers because we're so expensive. You're going to try to get every answer you possibly can. And those trials are underway. I can stop there. And
I think we're going to switch computers. Just see if we can make this work. Wonderful. Thank you so much for being here. I'm really happy to have this opportunity. So we're going to move into the brain now. I'm a neurobiologist, and I sort of unexpectedly got myself into this area of neuroimmunology. Now, it's been long known that the nervous system and the brain and the immune system interact on many levels in health and disease. And we know, for the large part, the brain is immune privileged. We have a blood-brain barrier that protects our brain against cells and other signals and proteins. But in the context of injury, damage, disease, this brain barrier can be disrupted, and this could cause a lot of problems in the brain. But today I want to focus on another aspect, which is an emerging area in the field, which is that our healthy brain actually consists of its own immune system. A large number of immune-related molecules that are typically associated with the peripheral immune system are actually expressed in the healthy brain by neurons and glial cells. And moreover, this cell, which is a cell called microglia, which is our resident macrophage that lives in our brain, These cells have an important role both in maintaining health in the brain, but also even acute perturbations local to their environment can disrupt their function. And I'm going to talk to you about what some of those functions are today and how understanding their normal biology can help provide new insight into new biomarkers and maybe even new therapeutics in the context of disease. So the wiring diagram of the brain is remarkably complex, as illustrated by this classic drawing by Ramon y Cajal. And as you can see, all of this complex signaling and connections between neurons in the brain. The human brain contains 100 billion neurons and trillions of synapses, which are the point of communication between neurons. And somehow each neuron receives these up to thousands of connections from another neuron, and that helps to form functional circuits that control very specific behaviors and thoughts. Now, the neurons are only half the brain. There's other cells in the brain. The other half is consisting of other cells called glial cells. And these cells, which also include microglia, are intimately associated with the neurons. So this is a synapse here, just a zoomed-in connection between two neurons. But you can see this intimate communication here between the microglia as well as other glial cells like the astrocyte. And what I want to highlight today is that this connection, even slight perturbations in the communication between these cells, can lead to really devastating contexts in the context of disease. So in the case of inflammation, even a local, very small perturbation in brain function can disrupt this communication. Now, microglia, what do we know about them? Well, we've known for a long time that they can both harm and protect the brain. They have very complex roles. They are, for example, dramatically changed in the context of injury. They go from this really beautiful process-bearing cell, and under the context of injury and disease, they undergo this dramatic change in morphology. They become phagocytic, as you heard earlier. They can engulf things. And we know they are the major contributors to neuroinflammation in the brain. Again, this is just within the brain. They can release cytokines and chemokines that can contribute to inflammation. But they're also good in that they can help clear pathogens and debris. And they also remove toxic proteins like beta amyloid in the context of Alzheimer's disease. So they're complex cells. And I'd say that one of the things that we've learned in the field is that we have to focus on the fact that there's not just one job of these cells, right? So they do these things like contribute to inflammation, and they can engulf things. But importantly, and what I want to highlight today, is these normal homeostatic physiological roles. If they're disrupted, this can have dramatic consequences in the way our brain and our synapses function. And connections between neurons and the synaptic function is critical to cognition and cognitive function, which is also an area that we need to be focused more on in the context of neurological disease. So it's important that we better understand microglia biology and their function and really understand the mechanisms by which these cells are disrupted in the context of disease. Now, one of the things that we have a challenge with, of course, is you can't just look into our own brains and study microglia. I wish we could. But fortunately, we have mice and animal models, and we can do things like transgenically label microglia with a reporter, meaning that all of the microglia in a mouse brain is conveniently labeled with this green fluorescent protein, for example, here. And we can actually take a mouse and we can just look under his skull, and we can put a microscope lens on top and watch the microglia in action in real time. And then we can ask questions like, what are they normally doing? And then how do they respond to different contexts of disease and injury? Now, this is an example of what microglia can do. So in these experiments, we can make a very local injury. This was classic experiments that were done and just replicated here by Lassa and Brian McVicker's lab a number of years ago. But I just want to illustrate the dynamic interactions of these cells. I'm going to try to play this movie. So inside the brain, you can make a little injury and look at the microglia go towards that injury site. So no other cell in the brain can do this. They're incredibly dynamic. Their processes are now moving towards the site of injury. So there are signals that are bringing them there, and then they're probably doing a combination of good and bad things. They're trying to help the injury, but in the meantime, if things get out of control, they can then release signals that make things worse. So I just want to sort of illustrate a sort of a dramatic example of what they can do. And you can imagine in the case of traumatic brain injury or something like that, this is what microglia might be doing. Now, they're also really good at eating things. They're phagocytes. They're our primary and our professional phagocyte in our brain. And this is another example, an experiment that was done by my colleague Axel Nimmeron, again using this live imaging approach where you can label the neurons red and the microglia green, and he just made a very local damage to that one neuron, and you can see what the microglia are doing. They're pulling up pieces of that dead cell. This is probably a good thing. You don't want to have this debris and dead cell around because that can actually lead to inflammation, right? So that's a potential protective signal. But you can also imagine if they are going awry, they could be engulfing too many things, which we think could also be contributing to disease and injury. But the thing that really got us excited as developmental neurobiologists is the fact that these cells are incredibly dynamic in normal conditions. They're constantly surveying the brain. These represent about 10% of the cells in our brain. They tile our brain. And even under normal conditions, you can see the fine processes of these cells constantly surveying the brain's parenchyma, the brain's environment. And even the most subtle changes, these cells respond to them. So they are, in many ways, well equipped to respond to both local and significant damage cues in the brain. So one of the things that we now appreciate that they're surveying in the brain are actually neurons and synapses. So this is just a zoomed in of a neuron in red. All those bumps are their synapses. And what you can see is they're constantly interacted with these synapses all the time. And this led us to wonder, could they actually be regulating synaptic function, right? And this, of course, is the basis of cognition, understanding the connections and the communication between neurons. Could these cells be playing a role? And what we discovered, and now many others have also shown, is that one of the things microglia are doing in normal development is actually phagocytosing these synapses. They're eating them. This is an example of a microglia, and all those little blue and red dots are the ends of synapses. And what we could see is during developmental critical periods where pruning is a normal thing, almost every microglia we surveyed were chock full of synapses. Now this, we discovered, was a normal role of microglia because pruning is a fundamentally important process that helps our brain wire up properly. So during development, we start out with an excess of synaptic connections, and through a process called pruning, a large number of these extra connections get permanently pruned away while others get strengthened and maintained, this idea of use it or lose it. And what we discovered was that microglia were one of the ways by which these neurons get pruned. And defects in this pruning process are thought to underlie neurodevelopmental and neuropsychiatric disorders. So pruning is really an important aspect, but synapse loss can be also a bad thing. You can imagine if too much pruning or not enough pruning is happening, this could have major consequences. And indeed, a hallmark of many brain disorders is the loss of synapses. This has been suggested in schizophrenia and autism in patients' brains and as well as in animal models. But it's also been very well studied in the context of normal aging and neurodegenerative diseases of the aged brain, such as Alzheimer's disease, which synapse loss is believed to be happening at a quite early process in the disease and is a stronger correlate of cognitive decline than anything else. So understanding what makes synapses vulnerable is really important. And we have now data to suggest that these cells called microglia are an important part of this process. So if we want to understand how synapses are vulnerable in disease, we again turn back to development, which is where we've been studying this process of normal pruning. And again, we have these model systems, mice, where we can really start to dig into the mechanism and try to understand this process. So the big question we asked a number of years ago is how is it that microglia know during normal development which synapse to prune and which ones not to prune? This is a cartoon that illustrates that early in development, a neuron is innervated by lots of inputs, and then you get a pruning of the blue and a strengthening and maintenance of the red. And we wondered, could microglia know which one? And if so, how do they know? Well, this was the other major surprise, is one of the ways that they know is through a group of immune molecules called complement. And these are molecules that are traditionally associated with the innate immune system that helps macrophages clear dead cells or debris. And what we found is, normally in the immune system, you would have these complement proteins tagging bacteria for elimination by macrophages. And what we found is in the healthy brain, one of the ways that microglia recognize and engulf these synapses is through complement. And if you knock out complement receptors or complement proteins, microglia are no longer able to phagocytose or engulf these synapses. So again, this is one of the many mechanisms, and we still have much to do to try to understand this. But in the brain, these complement proteins are normally targeting synapses. So we've spent a lot of time, and we still have much to do to try to better understand all the mechanisms that regulate this process. But we think it's important to do so because more evidence from my lab and now others suggests that this normal pruning process by microglia are aberrantly activated in a host of neurological diseases. The idea that if this process is slightly off kilter, meaning it's overactivated, this could lead to too much synapse loss. And this, we think, may be a process that underlies a cognitive dysfunction. And indeed, one of the big question marks in the field, and we know a hallmark of almost every neurological and neurodegenerative disease, is activation of glial cells. But the question is, when do they become functional? We can't know that in the human brain very carefully. When do they actually contribute to synaptic and cognitive impairment, and if so, how? Again, this is where animal models can be really powerful. So we know, as I mentioned, that synapse loss is the strongest correlate of cognitive decline. And so we decided to focus on Alzheimer's disease as our first step to test some of these hypotheses. The hypothesis being that this normal pruning mechanism gets aberrantly activated to contribute to synapse loss and disease. Now we know that synapse loss is actually a very early event in Alzheimer's disease, as I mentioned. And now the question is, are microglia in complement, could they be involved in this early process? And if so, could they be a pathway that we could start to think about targeting? And we recently showed in several different animal models that this pruning process, meaning complement deposition at synapses, now too many synapses, leads to microglia over-engulfing too many synapses in animal models of Alzheimer's disease. And now that's the big question that we're still now working towards, is we want to know, could we block this pathway and protect the synapses? And could that then protect some of the memory loss and cognitive decline? And so we have now started a number of experiments through collaborations with local Harvard Medical School investigators, Soyeon Hong, Dennis Selko, and our collaborator Cindy Lemire, where we used animal models and we knocked out complement or the receptors on microglia in these different animal models of Alzheimer's, and we asked, can we protect the synapses? And if so, can we slow the progression or protect against some of the memory or cognitive impairment? And this is just an example to show that we can quantify, count all those synapses, these are the yellow puncta, and we could show that getting rid of complement C3, for example, led to significantly more synapses in these mice. And quite excitingly, this also protected against some of the memory or cognitive impairments in these animal models. So this is just one example that I wanted to highlight today of a new role for microglia, independent of classic overt inflammation, that involves this synaptic dysfunction. And we think that if we could better understand how this is working and develop new approaches to try to detect this, this also might lead to new biomarkers in disease. And quite importantly, emerging genetics continually point to microglia. A lot of the genes, risk genes, almost half of them actually, are expressed in microglia or immune-related. And so that is telling us that it's not just a secondary effect, that these cells are critical and causal to the disease. So we need to better understand mechanisms. And I highlighted one for you today, but we have much more work to do in the field. So the other thing I wanted to say is I mentioned Alzheimer's disease as an example. And we, for many reasons, decided to start there. But we've now since gone on and looked in other models of disease, and others and colleagues in the field have done the same. And this recent work now suggests that this pathway I described to you today may be a common mechanism of synapse loss and cognitive dysfunction in a host of diseases. So Alzheimer's disease, frontotemporal dementia, glaucoma, and more recently, actually, there's even this West Nile virus can also lead to a cognitive impairment, and there's some evidence that this pathway is also aberrantly activated. So this is exciting because it suggests that even all of these diseases are so different from one another, but there may be a common denominator downstream that if we can find ways to perturb it, this might lead to new therapeutic approaches. And we're also now working to try to translate what we've learned in mice into human disease by looking in human brains and also thinking about that the complement in these microglia mechanisms might also represent an early biomarker for synaptic dysfunction. Because one of the things we're lacking desperately is a way to know when our synapses stop working. There's not a biomarker for it. There's not a PET tracer right now that's easily able to tell us if suddenly our connections, our synapses are starting to fail. So we think that maybe microglia might hold one of the clues, because they may be able to hone us in on and be able to sort of be a proxy for synaptic dysfunction in addition to their therapeutic potential. So I just want to thank everybody, and I'm happy to take questions in the next session. Thank you.
Take a seat now.
Oh, Diane's going to be there. She'll moderate. Okay. I was wondering about the chair scenario.
There's a musical chair.
Yeah.
So thank you for your many questions. I tried to put them together so we don't have duplication, and we sort of spread around questions for the various speakers. So the first question is actually for Steve, and that is, what are cytokines and their purpose within the body besides being used as markers for inflammation?
Yes, so they're mediators of inflammation. Cytokines are produced by immune cells and other types of cells, and they can promote other cells, activate other cells, and call them into activity at a site of inflammation. So they participate in the project. Substances produced by the cells and secreted into the local environment.
Okay, next question is there a way to force the displaced cells to disperse from the vessel walls once they settle there? That's for Phil.
Right, so there is a lot of discussion in the scientific literature as to whether you can force them out. So the idea of inducing a directional movement of the cells out through that outer layer, the adventitia, into the lymphatics, so where the lymph flows through as a different type of circulation. And it's difficult to know whether it would be possible in one respect, because as these cells eat all those lipids, they get really foamy, and it's difficult for them to move, right? So they stay put. They're sessile cells. They cease to be migratory cells. So how do you push a cell that's just a blob, a mass of fat? And so you would have to think about first educating the cells to be able to maybe get rid of that excess cholesterol that it ate so that it can start to move again, and then try to induce a directional program, it's called chemotaxis, for these cells to leave the lesions.
Okay, thank you. This is a general question. What kind of damage can long-term inflammation do to your tissues? Beth, why don't you start on that?
Well, I think there's certainly differences in the response of the brain to an acute damage. And then the chronic, actually, what was, I think, quite important to note is that when you get an acute change, that can completely change these cells, right? Even though they may not look different, a second insult later can then have a much more robust effect in the brain. And so chronic inflammation can lead to things like neurodegeneration and loss of neurons, loss of synapses, and a lot of other things, including sort of a cycling event. So that you have to really sort of think about, when you're looking and what the impact is. However, there's also protective roles. So some of the things that happens in the brain is that you get these cells trying to repair. You saw an example of that movie, where in many ways, if you think about it, microglia are probably in there trying to clear these things. But when the normal checks and balances go awry, it's hard for even the best efforts of these cells to not be able to start to go out of control. And I think that's the issue with these chronic diseases, is that these are things that are going on over many, many years, and often a second challenge or a second hit, whether that be genetic or an insult or an injury or a stroke, that can set off a cascade of events. And I think in the context of Alzheimer's disease, there's been some evidence of, as you may have heard, a traumatic brain injury or some sort of a second hit can then increase the progression and the onset of some of these diseases. So there's got to be a connection there. And it's more than likely because that first insult changed the way these cells like microglia are and therefore are going to respond differently to a second insult.
Steve.
I was just going to add a brief comment about a different tissue, and that liver also undergoes inflammation under a variety of different reasons, but just obesity is one, alcohol is another, hepatitis. And in that case, the inflammation progresses to fibrosis and cirrhosis as a direct consequence of the long-term inflammation. So there it's very easy to see and even grade in time the various aspects of it.
Phil.
And just another comment. So imagine what happens when you get an infection, right? When you get bacterial infection, right? The immune system uses an entire armamentarium to get rid of that infection, to get rid of the bacteria. And a lot of those things that it uses are very potent things. And a lot of these processes that we were speaking about here operate in the context of atherosclerosis or Alzheimer's. There's a collateral damage that occurs when these cells are responding with molecules that are very potent, that have, in some ways, have evolved to kill bacteria, but in doing so, in unleashing these molecules, they also cause tissue damage.
Thank you. This next question, since I know they were neighbors at some point, is definitely for Steve. Is there any actual benefit to Tom Brady's anti-inflammatory diet?
So I don't know his specific diet, but you can modulate your systemic degree of measurable inflammation by looking at markers by dieting, and that's losing weight. So if his diet is helping him to lose a little weight, and he's obviously in very good physical health, he probably has very low inflammation. I don't know if that's why he plays so well.
Thank you. Phil, what are the clinical trials designed to do for atherosclerosis?
There is one clinical trial underway. I think Steve mentioned this. This is a trial called CANTOS, and it is a trial that is targeting an inflammatory molecule, interleukin-1. And so it's inhibiting that molecule with an antibody. And it's a very large trial. There are many people that are enrolled in this trial. And it's asking the simple question, if you inhibit an inflammatory molecule, will there be benefit in terms of deaths from cardiovascular disease? The answer to this question will come within the next year or so. But I would just want to mention that regardless of what the answer is, this is just one molecule, one inflammatory molecule. If it succeeds, this will be an amazing success and certainly an amazing success story in terms of the role of inflammation in cardiovascular disease. But if it fails, we have to keep on thinking that inflammation, many of these processes in inflammation are really there are multiple things that are released. And IL-1, which is a cytokine, is one of those important but not exclusive things.
Thank you. This is for Beth. Can people with depression and anxiety or mood disorders have a lowered immune system?
Have a lower immune system?
Lowered.
Well, there's a lot of interest in research now in the context of mechanisms of anxiety and trying to connect that with the immune system. And I'd say, honestly, there's still a lot to be learned. We don't really understand it. But there is evidence of immune perturbations or models where you induce inflammation, even peripherally, that can, at least in animal models, lead to a more behaviors that are related to anxiety, for example. And that raises this question, what is the mechanism by which a change in the periphery could be altering the brain, and how that could be altering not just a particular behavior, but something as complex as mood, right? So I would say we're just at the very beginning of our understanding of how this works. And the big challenge, and the reason why we don't know more, is unlike the circuits that control like movement or particular behaviors, the circuits that control anxiety and higher cognitive functions like that are still not really well described. So if I were to ask the question, how does the immune system or inflammation potentially contribute? I would need to know what circuit or what part of the brain or how to model that. And we don't really currently have very good models. It's not to say there isn't research going on, there is, but I think that's one of the challenges, because a lot of these neuropsychiatric and neurodevelopmental disorders that don't involve one specific part of the brain, it involves the way you think, right? It involves emotions and feelings and social behavior. A lot of the effort in neuroscience now is to try to understand the circuits that control those behaviors. And we are really making rapid progress, but I think we need to know that before we can try to understand how the immune system influences it.
Thank you. So how does the discovery of crosstalk between intestinal microbes and the immune system change how we think about autoimmune diseases? I'm actually going to answer that myself because that's what my lab works on.
Yeah, good.
So we now know that the intestinal microbes are very important for maturation of the immune system. We know that for two major reasons. So one is that newborn people or mice have poorly developed immune systems until their intestine gets colonized with its natural microbes. And we also know it because experimentally we can make mice which are germ-free, entirely germ-free. And they have very poorly developed immune systems. And we can introduce then a constellation of microbes, and the immune system will develop. In fact, what we can do is we can introduce individual microbes, and that will steer the immune system to be either pro-inflammatory, have a pro-inflammatory flavor, or have an anti-inflammatory flavor. So the idea is that according to the exact composition of intestinal microbes, that may determine whether somebody is prone to autoimmune diseases or not prone to autoimmune diseases. It's certainly not the only factor. Genetics are important, and there are several other factors that are important, but it is a factor which can influence whether autoimmune disease develops or how rapidly or how severely it develops. And actually, you can't imagine how many biotech startups there are in Boston whose goal is to define either individual microbes or individual microbial products that can modulate the immune system for cancer or autoimmune disease treatment. Okay, this one is for Steve. What type of inflammation is the C-reactive protein testing? Is it chronic or acute or something else?
C-reactive protein, CRP.
Yes.
So we know that it goes up in many kinds of inflammation. We don't actually know what the function of that protein is. So that would be considered to be a marker, and we don't know that it's a mediator because we don't have an ascribed function for it. So when it's up, there's some form of inflammation somewhere in the body, and when it goes back down, that has likely resolved, is what we use that as a marker for.
Thank you. Phil, is there an increase in cholesterol caused by stress or anxious state, the emotional state?
Is there an increase in cholesterol?
Cholesterol.
That's a difficult one to answer. Certainly there is an association with stress and eating. So whether there is a direct link between being stressed and having cholesterol, independent of the behavior that stress can cause, that's an interesting question.
But I think that actually really nicely illustrates the fact that there's communication both from the periphery, the immune system, to the brain. And this might be an example where things that are happening in the brain control the immune system. And I think there's evidence on both sides, but not enough known on how that might work. So it's an interesting idea.
Yeah, that's good.
Okay, here's one from social media, and it can be attacked by any of you. At what moment in the cascade of inflammation must we act?
One thing to think about, maybe I'll start. One thing to think about is that one perspective here is that there's always inflammation. We're always, inflammation is within us at all times. It's just the question is the balance. And too much of something, we're always walking at a knife's edge. When I was showing you the production of those monocytes and neutrophils in the bone marrow, they're being produced at all the time. It's not just when there is a danger, where there is an infection or an injury. So that's, I think, a good maybe place to start, that we are sort of walking inflammatory organisms, and what we don't want is the teeter to fall into overt inflammation. Maybe that's...
Anybody else want to say anything? Okay, Beth, what's currently known about insults to microglia that may lead to gliomas?
Yeah, I think there's emerging evidence that implicates these cells in glioma. They certainly are going to be responding because there's now a local damage or tumor that releases a lot of these cytokines. And they're also phagocytic cells, so that they may help clear some of these things. But I guess there's still not a lot known mechanistically how they contribute either positively and negatively to glioma. I know this is an emerging area. We just recently organized a conference where this was an emerging theme. So I would say stay tuned for that. But these are, just like I mentioned, all of these cells are intimately associated with one another, and perturbations in the environment is going to basically alter microglia's normal functions. So in one context, you can imagine just inflammatory signals or signals from these tumors are going to change their state. So in one way, again, protective potentially, but also it could lead to, if there's not breaks in place, it could lead to their inability to normally do their normal day jobs. And I think that this is another thing in these animal models, this might be helpful, because I think there's clear evidence that they're activated in the case of glioma, but what they're doing and how they're contributing is still an open question.
Thank you. Steve, this is also one from social media. Is there a hypothesis for the mechanism by which IL-1 beta inhibition reduces hyperglycemia?
There are hypotheses. So that when we put cytokines like IL-1 beta or TNF-alpha onto even cultured cells, they become less responsive. So there are proteins activated in the cell, and there's crosstalk between pathways. So the cytokine has a signaling pathway in the cell, and insulin has a signaling pathway in the cell, and the two speak to one another. So that when the cytokine pathway gets activated, the insulin pathway doesn't respond as well anymore. So that's, in a simplified way, that is one explanation of how it can occur. There are other explanations too, and we're trying to get to the bottom of it.
Thank you. Phil, is gout still considered an inflammatory disease? Are there any new treatments?
Well, it's uric acid and IL-1. I mean, this is another disease where IL-1 is very much a culprit here. So yes, the answer is yes. I mean, it can be treated very well with anti-IL-1 inhibitors. It can be treated. So in that respect, it's a success story for how inhibiting inflammation treats gout. But it is a disease that has a major inflammatory component.
Beth, maybe you can shed light on this one. Does inflammation contribute to seizure disorders like epilepsy?
Yeah, it comes back down to that same thing I keep raising, this sort of cause versus effect. Again, this question of, when you induce a seizure, and this can be, again, modeled, dramatic changes in these cells, right? And this leads to inflammation and an increase in a lot of these immune molecules that I was just telling you about. But the question of whether they're sort of responding to or contributing to has been really hard to parse out. So I think what we need to do, and the strategy that I think many are starting to take, is, again, using animal models, you can start to ask, if you want to know chicken or the egg question, you need to be able to specifically block microglia or some pathway in them and ask how that contributes to things like epilepsy or seizure. And we finally have tools in the field where we can genetically either manipulate just those cells and no other cells in the entire body or brain and ask how that impacts things like the seizure progression. Or you can actually monitor them with these different reporters. So I think that's what we actually need to do, but I don't think that anyone's really been able to parse out the contribution versus the response at this stage.
Thank you. Steve, considering all macrophages' actions, do they have an effect on obesity?
So macrophages, if the question is, will macrophages reduce the degree of obesity? I doubt it. I think they're responding to the obesity because there's more of them. We're trying to find out exactly why there are more of them. We think it's in part due to tissue remodeling. After all, the adipose tissue is able to change its size and shape more than almost any other organ of the body, perhaps besides female sex organs, that can change very dramatically and rapidly with time. But we can take on many, many pounds and then shrink back down again. And that's historically been very necessary to survive periods of famine. We no longer have to survive periods of famine, so we only have the other side of that coin, and we just gain. And we don't lose, and then we gain a little more, and we don't lose so well, and we gain some more oftentimes. And the macrophages are needed for all of that because as tissues grow, they need to change their ability to expand, remodel, and the macrophages are the main mediators of remodeling.
Thank you. This is a general question. What nutrients can reduce or prevent chronic inflammation? Who would like to take that on? I have a general answer. All the foods that you don't like, they're anti-inflammatory. Broccoli, cauliflower, all that stuff.
I would add that probably eating a little bit less of each thing is the surest way of having a little less inflammation systemically because you're going to lose some weight, and those two do go hand in hand.
Thank you. Beth, does the inhibition of C3 allow the brain to repair itself in some way, or is the damage irreversible?
Well, right now we think the inhibition of C3 is, if you catch it in time, early enough, it can prevent the subsequent loss of synapses and damage to the brain. At least the data thus far suggests that by knocking it out, it's reparative. However, it's not known yet, and we haven't done this experiment, is if the disease has progressed too far along and then you inhibit complement, does that help? Does that slow the progression more? Those are experiments that are now in progress. So I think that's actually going to be really important to answer that question, not just in the context of Alzheimer's, but to other disease models as well. When is the right window of intervention, right? And I think, my opinion is that one of the reasons why a lot of the clinical trials have failed in a lot of these neurodegenerative diseases, including Alzheimer's, is that we're not going in early enough. We don't yet know when to go, especially when we don't have biomarkers and we don't yet know who's going to go on and develop Alzheimer's disease. So the hope is that if we could identify a critical period as window where it's still helpful, that would be hugely helpful. But at the same time, sometimes we don't have a choice, and we want to know if we go in much later, can we still be protective? That said, what I think the other evidence in the field suggests is that these complement molecules, that we are probably only at the tip of the iceberg of understanding their normal functions in the brain. This is one of the things they do, eliminate things, but there's emerging evidence that suggests that they have a lot of other roles in the brain as well, which is another potentially interesting but problematic aspect in terms of therapeutics, because you don't want to just block complement everywhere because they have a lot of good functions. So one of the strategies that we're taking is to try to understand mechanism of how complement in this context is contributing to synapse loss, and if you can then design either small molecule or very specific ways to disrupt just that process without disrupting all the good things it does, this might be a way of intervening with in a very specific targeted approach. So I think this whole immune molecules in the brain, all very exciting, is also very complex. And we need to start thinking about, again, understanding deeply the mechanisms and the biology to try to develop new strategies to intervene at the right time.
Thank you. Phil, how are anti-inflammatory meds harmful if taken for too long?
Well, so this harkens a little back to a point that Beth just mentioned, and it's that the immune system is there to protect us, and we're in this constant, if you will, a balance in the best of times between agents that want to harm us and those that will protect us. Now, even when you bring in something like the microbiome, you've got different species, you've got the bacteria that can be very beneficial, but if you allow them to enter into locations where they're not supposed to be, that may be a very bad thing indeed. So you never want to turn off the immune system, and I think that is one major challenge in thinking about use of anti-inflammatories, because you want to give the immune system the opportunity to react when there is something truly scary that infects or strikes. And so if you are giving a lot of anti-inflammatories and end up shutting the immune system down or preventing it from doing its normal job, you may be very susceptible to infections. And this is, in fact, something that happens quite a bit in the hospital with nosocomial infections that occur in sepsis patients whose immune system is so overactive at the beginning of sepsis, where they have this overwhelming infection and an overwhelming reaction to an overwhelming infection, that essentially these immune systems shut down. They're just overheated, to use, for the lack of a better word. They can't operate or function very well anymore, and that is a great opportunity for a bug that would like to exploit that.
Thank you. So even though we have at least a dozen more questions, we only have time for one more, unfortunately, and that's for Steve, and that's looking at the other side of the coin, I think. Could you speak to the studies indicating that doctor-supervised extreme caloric reduction can eliminate type 2 diabetes?
Well, it does decrease. So caloric restriction is a beneficial thing to do. We have the most experience in doing it with animals. We have lots of data showing that it not only decreases levels of many of the different diseases that we've been discussing, but it also increases longevity. The animals live longer. So it sounds great. People have also, there's a community of caloric restricting people that believe in this. The problem is that you're not in a very good mood when you're eating 70% of your normal diet. The people are grumpy all the time. You're just not living a good life anymore. So you can't drop it 30%. It's a typical amount, 30% reduction, and that isn't very happy. So dropping it a little bit less than that, maybe dropping it 10%, would be a much better approach to it. But that isn't called caloric restriction. That's called dieting.
Okay, thank you very much.
Yep.
Thank you.
That was fun.
That's fun.
Good answer. Yeah, it was quite impressive.